Hi, I'm Alan Snavely, a junior molecular biology major, and welcome to my Made in Millersville presentation. The title of my presentation is Game of Microbes, and the core question behind my research has been trying to answer the question, which microbes are present in hemp, and what does this mean for potential crop rotations in Pennsylvania? Hemp is cannabis sativa, although the taxonomy behind its name has been subject of many botanical arguments, with some suggesting that cannabis indica is indeed its own species. The general consensus is that indica is a subspecies of sativa. The line between marijuana and hemp is the level of delta-9 tetrahydrocannabinol, or THC, which is the compound that gets you high. Hemp has less than 0.3% THC. Anything greater is illicit marijuana and must be destroyed. Hemp is booming in popularity due to its many useful products such as seed, fiber, and cannabidiol, CBD, which is great for anxiety, and I could use some. Hemp has become the next big cash crop and is the highest grossing income crop a farmer can plant. Nationwide, hemp has grown to be a $100 million industry. In Pennsylvania alone, the Pennsylvania Department of Agriculture released 510 industrial hemp growing permits, 94 of which were granted to Lancastrian farms. But we don't know how to grow hemp. Since the banning of cannabis in the 1930s, hemp production and the knowledge of how to grow it has stagnated. We don't really know much about it. Crop rotation probably brings a picture to mind, at least if you're from Pennsylvania. It's such a commonplace practice. In fact, it's one of the oldest agricultural rules of thumb known to humankind. The basic principle is to plant a different crop each season. In doing so, nutrient usage will be optimized, soil quality will improve, and disease will be prevented. But why? In the case of nutrients, different plants need different nutrients. Looking at the figure on the right, in season 1, nitrogen-fixing plants like soybeans are grown to increase levels of nitrogen. The next year, a crop is grown that likes the extra nitrogen, and finally, a light feeder is grown that does not need much nitrogen. So, season 1, something like soybeans would be grown to fix nitrogen. Season 2, something like corn would be grown that loves the extra nitrogen. And then season 3, either carrots, onions, or even hemp could be grown as their light feeders. Soil quality is a less understood principle of crop rotation, but the general idea is that certain plants' root architecture improves characteristics of the soil, such as aeration, porosity, and particle clumping. Recent research shows that hemp root architecture is conducive to improved soil porosity and quality, further supporting hemp's place in a crop rotation system. The final consideration for a crop rotation system is disease. For disease, less related plants will have less shared diseases. I don't know if you've noticed, but your pet cat isn't getting sick from your pet goldfish. And it's the same for plants. If corn is planted one season followed by soybeans, the diseases of each crop will not be transmitted. But if there's no rotation, diseases will be transmitted year to year. Hemp, corn, and soybeans are an excellent example of a perfect crop rotation system in the context of disease. Corn is a grass, a graminoid. Hemp and soybeans are both eudicots, so they're a bit more closely related, but they're also in different orders and families. These plants are not closely related and should make an ideal crop rotation system. According to Dr. Alyssa Collins of Penn State, Pennsylvania hemp growers have been employing the soybean-corn hemp rotation model. This model fits the nutrient usage rules outlined in the figure, the relatedness rules for disease, and might even fit the soil. According to recent research, the magic behind crop rotation systems stems from the microbial communities. These communities occur all over nature, from your intestine to the ocean floor. These communities are not inert, but are actively communicating, competing, working together, and stabbing each other in the back. Like Cersei says, when you play the game of microbes, you win or you die. The diagram on the right outlines these communities in plants. Microbial communities are made up of fungi, protists, viruses, nematodes, and bacteria. Bacteria is the focus of this presentation. These communities affect nearly all plant processes, from nutrient sequestration, hormone production, disease prevention, and causation. An example of nutrient sequestration, or simply nutrient gathering, is an association between soybeans and rhizobacteria. Soybeans give the bacteria carbon, while the bacteria supply the plant with nitrogen. The diagram on the right shows that these microbes are actively communicating with each other and the plant. They're fighting for their microhabitat that they're best suited for, which is called a niche. These microhabitats might be differences in moisture or pH, and the sheer number of these niches present around the roots are what allows these communities to be the most diverse on Earth. Utilizing this information, we've hypothesized that corn, soybeans, and hemp will share key bacteria that may impact a potential crop rotation system. So bacterial communities are of so much importance, but how do we measure them? How do we take soil and turn it into data? The first step of our methods, and perhaps the most important step, is sampling. The first step of sampling is choosing a sampling site with known abiotic factors. Abiotic factors are environmental factors like temperature, precipitation, or land use factors like tillage and pesticides. At Penn State's research farm, we had all these factors accounted for. So the next step of sampling is to take consistent amounts of soil from around the root at consistent depths from randomized plants of the same cultivar. Once we have our soil samples, we take the soil back to the lab and make a beautiful microbial soup. With this soup, we start diluting it, as you can see in the far left picture. Once we have our dilutions, we want to isolate individual colonies. An individual colony, like the one here in the middle plate, the big yellow dot, is then further purified till it looks like something here on the far right. As you can see in the middle plate, there's quite a lot of contamination. There's 3 or 4 species of bacteria and fungi here, and this is why this process takes so long until all of our isolates look something like this. Over the course of about 3 months, we've been able to isolate about 62 bacterial strains from 7 cultivars of hemp and 1 cultivar each of soybean and corn. As you can see on the table on the right, we have varying numbers of isolates from each sample, and not all samples we took produced isolates. Below, you can see a picture of all of our isolates, and you have to appreciate that because it took a really long time. The next step of our methods is what we are doing currently: bacterial purification and gel electrophoresis. So once we have our pure bacterial strain from step 2, we must isolate the DNA. So to isolate the DNA, we basically just break the cell open, we denature it. And then once we have the DNA, we must amplify it using RET-PCR, or repetitive palindromic polymerase chain reaction, or RET-PCR. We actually didn't get this far with our research. The RET-PCR step did not work for us in the lab, as soil bacteria is highly resistant to change in abiotic factors like temperature. And RET-PCR uses temperature to destroy DNA and then lowers temperature to reanneal it. So if RET-PCR were to work for us, we would visualize the differences between the strains using gel electrophoresis, which you can see on the right. If bacterial strains were the same, they would share similar banding patterns. So looking at this culture here and this one, they have very similar banding patterns, so they're most likely the same organism. Same for these two. So if samples share highly similar banding patterns, they're most likely the same bacteria, or at least very closely related. So far, I have been outlining the culturing approach, but a more modern approach is that of the microbiome. The difference between microbiome work and culturing the microbial community is that the microbiome accounts for more than just the culturable bacteria. Sorry, I mean culturable microbes. And by culturable microbes, I mean one that's grown in the lab on an agar dish. The microbiome represents all bacteria, all fungi, all viruses, regardless of if they can be cultured or not. So how do we identify the microbiome? Steps are similar to our past methods. First, samples are collected exactly like they were before, but instead of making a microbial soup, we go right to the sequencing step. So once DNA is attained, it's sequenced using 16S rRNA for bacteria and ITS for fungi, and then the sequences will be processed using the R program. So the microbiome approach is our next step in research, and it should definitely illuminate potential interactions in a corn, hemp, soybean crop rotation system. I'd like to acknowledge Melissa Gold and the rest of the McNary Library Fellows. They've really supported this work throughout the pandemic, and I don't think I'd be here without them. I'd also like to shout out Dr. Ramos Sepulveda. She's the best professor in the bio department and my research mentor behind this project. I'd also like to thank the people behind the Muse grant and the SGRCA for helping to make this research possible. And I'd like to thank you for listening.